Okay, good afternoon, everyone. Can you hear me?
Sure can.
I
Okay,
can hear
great.
you.
Yep.
Great. Thanks, everyone.
I can
Okay,
hear you.
great. Welcome this afternoon to the Medical Marijuana Testing Advisory Council meeting. We'll be going to approximately 2:30 today. We've got a packed agenda. I'll just introduce myself. I'm Peter Waddell, Bureau Chief with the State Public Health Lab here at Department of Health Services. And I will be filling in and taking over this position. As many of you might have heard, Jessica Rigler has moved on to a different role outside the agency. So I'll be filling in this position going forward. So I just want to go forward this time and want to do a quick roll call of all of the folks that are attending and are council members, and I will call out your name, and if you could please let me know that you're there. I will check you off on the list. I'm going to start with Torres.
I'm here, thank you.
Great, thanks, Anne. Ryan Tracy.
Present.
Thanks, Ryan. Steve Cottrell.
Present.
Thanks, Steve. Murray Stein.
Here.
Thanks, Murray. Matt Lascala.
Hello, I'm here.
Matt. Andrew Provicino.
It's Andrew Provencio and I'm here.
Oh, sorry, Provincial, sorry. Tabitha Muir.
I'm here.
Tabitha. Hope Jones.
Hi, everyone here.
Thanks, Hope. Joe Demena.
Here.
Thanks, Joe. Jay Neary.
Present.
Thanks, Jay. Vince Figueroa.
Here.
Jean, if I got that right, sorry if I got it wrong.
I'm here. It's pretty close. 中国熊.
Okay, thank you. Maxwell Nunn.
Yeah.
Thank you. Alvin Altamarino. No, last call for Alvin. No, I don't see Alvin, okay. Lastly, Ty Garber. Okay, Alvin, we see you there, great, we'll check you as present, thank you. And lastly, Ty Garber. Is Ty there? Okay, we don't see Ty. So we have everyone that's expected. We still have one vacancy on the council at this point, which is for the licensed healthcare provider. So we'll still work towards filling that. Again, thanks everyone for joining today. I believe we've got a quorum, so we can kick off the meeting and we can move forward. So I've just got a few comments here before we start to get into some of the topics that are on the agenda. Again, thanks for everyone for attending and for submitting items for discussion as well. Just a reminder of the open meeting rules that we follow is when speaking, council members should identify themselves for clarity. We may also only discuss topics that are on the agenda today. So if something comes up, then we can consider that for a future agenda, but can't discuss it on today's call. And also today's meeting ends, as I mentioned earlier, at 2:30 p.m. So if we don't have enough time for all of the items on the agenda, we can add them to a future agenda for one of our future meetings. And again, the council must stay within the bounds of their statutory authority. So I thought it'd be good to just quickly check over the council's charge at this point in time, just to remind everyone what we're charged with as a council. So the Medical Marijuana Testing Advisory Council shall annually review the marijuana testing program and submit a report by the second Monday in January. And this report should include an assessment as to whether an analyte should be removed from the required statutory testing panel. The number of statements of deficiencies should also be included in this report relating to testing deficiencies that were issued to each nonprofit medical marijuana dispensary, third-party independent lab, marijuana establishment testing facility in the preceding year. The remediation efforts made to address each deficiency and the resolution of each statement of deficiency. And also, if there are any other recommendations of how we might improve the testing program, those are also items that we should include in this annual report. And so just as a reminder, we had our last report that was due this January. Was submitted, so the department has been continuing to work towards the goals in that report, have regular stakeholder meetings to provide technical assistance, and also to discuss topics such as packaging and labeling, the recall process, definition of a batch, and also certificates of analysis. The department is undergoing rulemaking, which will address some of the feedback received from the MMTAC group. And Steve Baker's presentation that you'll hear soon will be covering some of those things in more detail. And so with that being said, we can move forward. Our first, I'm going to turn it over to Megan Whitby. Let her introduce herself, and she will be presenting with Steve Baker on dispensary establishment deficiencies, and then Steve will present on lab deficiencies. Megan?
Thank you, Victor. My name is Megan Whitby. I am the Bureau Chief for Special Licensing. So my team oversees the dispensary and establishment licensing, as well as all of their related facilities. And can we see the testing deficiencies, please, on the screen? It has a blue line at the top. Just for the panel, for the council members and everyone, these testing deficiencies will be updated. There is every meeting until we actually have our final report, which will be due in January. So by the time we submit that report in January, we'll have the full year's worth of testing related deficiencies. But just an overview today of what we have so far this year. And just for clarity, these are all of the deficiencies that have been completely wrapped up, meaning the inspection's taken place, and we've gone through the whole process through to accepting a plan of correction. So at this time, we have cited 24 different medical marijuana dispensaries for testing deficiencies. Those were done through 26 unique statements of deficiencies, which would mean we have 26 different inspections of those medical marijuana dispensaries. We had 163 citations on those medical marijuana dispensaries, but I do want to put that in a little bit of perspective for you. All but 5 of those were related to policies and procedures regarding testing. So on the dispensary and establishment side, we really tried to make sure we include every possible testing related deficiency, even if it's kind of a tangential relationship to testing there. So the vast majority on the dispensary side, the citations have been due to issues with their policies and procedures, which can result in multiple deficiencies on a statement of deficiency. On the establishment side, we have cited 38 different adult use establishments for testing deficiencies. Those came to a total of 40 statements of deficiencies. So that would have been 40 different inspections, and for a total of 63 citations related to testing on the adult use establishment side. So again, if we have anything in process where we haven't had a plan of correction completely accepted, or if it's possibly in enforcement right now and we're still going through that process, we have not reported that out here, but we will continue to update this throughout the year until we have the full picture in January for the report. And I'm going to turn it, unless there's questions, I'm going to go ahead and turn it over to Steve Baker for the lab side.
I have a question.
Sure.
Is it possible to identify each business that is responsible for these infractions, i.e. each dispensary that shows those? Right now, it just identifies dispensary D106. Can we?
So for the report itself, they are supposed to be de-identified. We can provide more information related to establishments, but because of the confidentiality statute for medical marijuana, we cannot do that on the medical marijuana dispensary side.
Correct, only for adult use.
Yes, and for adult use, just so everybody knows, all of the statements of deficiencies for any of these, they're already online through AZ Care Check. So any deficiencies for any of the adult use establishments are all deficiencies, not just testing related deficiencies. They're all available on www.azcarecheck.com.
Thank you, Megan.
This is Ryan Tracy with C4 Labs. I had a quick question.
Sure.
I was just wondering, do you have a breakdown or even just a general summary of what the common violations may have been? What was seen the most out there in the field, kind of identifying where maybe some of the lack of communication or just lack of understanding might be present?
Like I said, for the dispensary side, it was largely issues with policies and procedures, but we can definitely take that feedback and incorporate it for next time and try and give you some hot topic areas or common violations that we're seeing in dispensaries and establishments, if that would be helpful.
Yeah, it may just help us discuss that to hopefully be able to remedy it here with the council.
We will work on that for our next meeting.
Okay, if there's no more questions for Megan, we'll turn it over to Steve Baker. Steve?
All right, well, thank you. I'm Steve Baker. I'm the Chief of the Office of Laboratory Licensure and Certification. And one of my programs is the marijuana lab licensing program. So can you bring up my presentation? So mine's a little bit different. I have it in the format that I had it last year at this point, and there was pending information. And we had said that we were going to allow it to be open for a point in time. So the pending issues that you see in there that have spaces are those similar to last year that have had a report of findings, but we still need other information. But I believe that all of it will be easily done by the end of the year. So it should be able to get it onto the report that goes to the legislature and the governor. So as you see, we have about 22 laboratories that are currently in the process with some kind of inspection that has occurred. We have about another 3 or 4 laboratories that are about to apply. So quite a few more, I believe, than last year. If you go to the top, you'll see that, again, we're not listing the labs by name, and they are not in alphabetic order. You might be able to tell whom they are. If you're the lab, I know a lot of the lab reports have gone out and are being reviewed, but anyways, most of the laboratories that had some kind of enforcement action going last year have had consent agreements and have paid civil money penalties. You can see that some of the laboratories that are in the process, we're recommending some civil money penalties, or we've already assessed notice of intent to assess civil money penalties. But the thing I'd like to everybody to know is the difference between last year's inspection and this year is pretty night and day. And that's a good thing that the council should know, is that you can see maybe even from lab number 3 there, how they had 60 deficiencies, and then this year they're on 26 deficiencies. All laboratories have far fewer violations. The civil money penalties that are being considered are far less this year. What the council should know is that I think I said it'd take about 2 to 5 years to get to a point in time where the labs are starting to get in pretty good shape, and I think we're getting there. So that's a good thing for the council to know. We have had some laboratories, as you can see in number 6, that have come on, have had issues, have withdrawn their application, and then have come back on. So if you keep going down, you'll see what the pending enforcement actions may be. But overall, a lot of laboratories, many more than I thought would be coming on. Maybe more laboratories than the market can hold and keep going. So that will be interesting. We currently do not have any adult use laboratories. I'm not sure why you would, because all medical marijuana labs can automatically test adult use products. So, and are there any questions?
Hey, Steve, this is Zhongguo Xiong. So I see some of the labs has like 2 rows and some have 3 rows, some have even 4 rows, 5 rows. What are those rows indicate?
Okay, well, some laboratories, when they apply for like approval of testing, well, each row is a separate inspection report. So you look at number 15 laboratory, there's 5 rows. That means we went out to that laboratory 5 different times. We have 5 different reports. So what happens is, it depends on the lab, really. Some laboratories come on board, they apply for all testing. And so you may only see, like in lab number 16, you know, with the 84, that could be for all of the tests. Whereas in lab 15, they're probably coming on test by test. So they may apply for heavy metals, then they might apply for micro, and so on. So when you look at the deficiency number, such as the 125 or the 84, that doesn't necessarily mean that the lab is any worse than the lab in 15. It just means that if you added up all the deficiencies in lab 15, it could easily, it could be the same thing at that point as if you add them all up. So it's really hard to get a sense sometimes I don't think you can look at the violations, because this also isn't saying the severity of the violation. You can have one violation that the lab is approved for testing, but is falsifying all the testing and not actually doing any testing. That's one big violation. So it's really hard looking at this chart to determine the severity of the situation, and necessarily you can't do a who's good or bad based on this chart, but it does kind of tell you what kind of enforcement actions are being performed, we're having to go through, and the fact that the violations are definitely going down.
Thank you, Steve.
Hi, this is Hope. I raised my hand for a question as well. So Steve and for the data that was presented right before Steve, I'm just curious what we're supposed to do with this other than, as you kind of just said at the end there, Steve, just to see that citations are going around. I'm not really sure how to think about these because I don't necessarily know exactly any detail other than what you guys are showing just right now. Is there, maybe can you provide a little bit of discussion as to, is it just to see that you guys are doing enforcement, I guess is my question?
I don't know.
Well, it's really specific to the Medical Marijuana Testing Advisory Council statute that was changed, not this legislative session, but the last one, where it dictated that we have this annual report. And it was very specific about the things that had to be included in that annual report. And this is the specific data that was required of the department to provide.
But I seem to recall last year that we were discussing having actual information in there that was more informative for us to think about as a council on what to do with some of the citations, or to Ryan Tracy's question earlier, helps us maybe understand how we might be able to word or approach some of the changes or discussions that we're making, so we can either minimize what some of these general citations might be. For example, if it's just something that we could target more directly.
So, and like I mentioned to Ryan, we can absolutely start looking at some trends and bringing that information to the advisory council meetings if that is helpful. But for as far as what has to be in the actual report, it's very specific in the statute, and so we have to stick to make sure the data is presented in the way that it's required by the statute. But for the purposes of these meetings, like I said, we will bring some of that trend data to the next meeting.
And Hope, as a workaround, being that we don't have the same protections as labs, you can do a public information request and actually get the physical copy of the inspection. So if there's operators or somebody out there that would like to go a little bit deeper than this to really inspect a lab or to see kind of where the deficiencies are, that is something that is available. And I hope that will keep labs honest.
And also for the establishments, you can, again, the medical marijuana ones are confidential, but the establishment statements of deficiencies for all of their deficiencies, including testing violations, are on Easy Care Check.
Okay, Audrey, let's see, if your hand raised, would you like to ask your question, please?
Was that to me?
Yes,
What'd you say again?
Andrew Provencio.
yes.
Yeah, so I just had a follow-up question on Hope's thought. Is there a way we could rank the violations, with, severe violations, having a number 3 and, so we could see the severe violations, because that would be a little bit more descriptive. Obviously, a file being in the wrong place, but causing an issue on violation versus somebody that falsified a test would be different severities of violation. That's just a thought, and that's.
So I think what we can do is definitely tell you which rules are being violated most often, which citations we're seeing the most of. As far as the scope or severity of it, I mean, I think that would be a whole other discussion on determining which violations went into which rating. So I don't know that we want to get into that just now, but we will definitely provide more information about what is being cited and how often they're being cited.
All right, thank you.
Anyone else? Any additional questions for Steve or Megan? Okay, I don't see any, so we will move on to the next agenda item, which is Steve Baker again. Steve, you're going to give us a review of the upcoming rule changes, please.
Some of them. So if you could bring up my PowerPoint. All right, so if you put it in, there you go. All right, so these are for the lab part. There are a couple here that are actually in the dispensary rule side, but we'll go over those. So these are what I consider to be some of the significant things that I think are significant. Others may think others are, but anyways, we'll get into it here. And again, we have already posted the proposed rules. We've been talking about this for quite a while. For the medical marijuana program, we have not done an update since January of 2021. So what we're doing now is we're entering into the regular rule process. And to do that, you first have to get an exemption from the governor's office, which we got. So we are now in a 9 to 12 month period where we send out draft rules, go through comments, have possibly public meetings. There's a whole process, go to GERC, the Governor's Regulatory Review Committee. So there's a whole process here. This is not going to just be happening tomorrow. So everybody, there's a chance for all of you to make comments, send those in, and discussion. So next slide.
Steve, can I interrupt you for 1 second? I'm sorry. This is Megan. I apologize. I just wanted to clarify because a lot of people on this call have been part of marijuana rulemakings, and this is very different than what you've seen over the past few years. So starting with SB 1494, which is what implemented the testing rules and everything, we were given super exempt rulemaking. And then followed up, we also had the Smart and Safe initiative that brought along adult use and also gave us super exempt rulemaking. And with that, we were able to fairly easily update our rules and not have to go through some of the regular processes that you normally have to for rulemaking. And we were able to see those rule changes implemented very, very quickly, like almost like within a week, sometimes we can do those. So this is not that process. And I also wanted to point out that we also have an expedited rulemaking that is impacting medical marijuana that we already went through all of the public comments on. There are a few changes to testing, but they were required by statute. They were taking out the one residual solvent, the residual solvents for flour, those types of things that were changed in statute last year we were able to change through that expedited rulemaking that will go into play much sooner than the one Steve's about to talk about. But the main part of that expedited rulemaking was updating our allocation rules in medical marijuana. So there will be some changes coming in the next few months. We just have to go before GERC, the Governor's Regulatory Review Council. We're almost there on those ones. And then these changes will be following that. And like Steve said, because this is a regular rulemaking, it's a much lengthier process. You will have plenty of opportunity as members of the public to comment on different drafts as they go through. But I just want to clarify, there is another rulemaking that will be taking effect for medical marijuana prior. Sorry.
And can you resize the screen because we can't see, I can't see it. It's only showing me half of your presentation, please.
Oh, because I can see the whole thing.
And just, they are available on our website as well. If anyone is.
All right. So I'll get into it. And so the first proposal from the department, if you remember from the fall of 2019, the proposal from the department was for the Escherichia coli to be 10 CFU per gram or lower. Most states have a standard of 10 or lower. And that's typical of food standards that are out there for most of the programs. However, I knew that Oregon had 100 and we were kind of on a roll for Oregon at 100. So the problem is when you have Escherichia coli or E. coli, as some of you hear out there, that means you have fecal contamination going on. So anytime you have fecal contamination in a product, then that means there's the potential to have pathogens that could cause harm. And 100 CFU per gram, which is much higher than any food safety program I'm aware of. 10 is a better standard for edible products. So what we're proposing is to split out edible marijuana products to cut the amount of fecal contamination. 100's not, well, 100 is high. If you're smoking marijuana, most of the E. coli is being burned up anyhow. And so I haven't gone back and looked to see at the states, but some states only regulate flour or different products. And as far as topicals, 100's probably not a problem. I mean, it's not a great thing to have fecal contamination, but anyway, that's one of the proposals that we're sending out for comment. The other comment thing that we're looking at as far as the standard is the mercury level. So there's only one other state that has a higher mercury level than ours, and it's not for inhalable. The problem with mercury is with inhalable. That's where most of the damage comes from, because it's a pretty quick damage to your brain and your nervous system. So all of the states have a lower standard for inhalable products because of that. And most of the states, the USP limit is 0.1. We're proposing 0.2. We have California has 0.1, Maryland or Michigan, Montana. Most of the states have the 0.1, but we had proposed originally to go with Maryland's and New York and some of the other states at 0.2 because of they were looking at it as more as 0.1 as the limit of detection, and then the safety limit is doubled. But anyways, so we're proposing the 0.2. The 1.2, if you remember, it was kind of pulled out in the fall of 2019 based on one laboratory's limit of detection. So it wasn't based on a safety limit. So anyways, that's what the proposal is that we're sending out to the states. Next slide, please. So we're making a couple of changes in the pesticides. The abamectin comes in, abamectin is B1A and B1B, I believe. The problem with B1B portion of it is extremely low. So we're proposing that the review that all the review that the laboratory has to do is for the B1A form of abamectin. As far as the pyrethrins, I think this is also a comment or a proposal that Tabitha was bringing up in her comments, is we're getting rid of the proposed getting rid of the cynarin and the jasmine because they're very minor components, but we are adding the pyrethrin 1 and 2. When we called up Oregon and asked them why they had left out the 2 component, they didn't know because there was no history in the program. So what makes sense is to do both of the pyrethrins and to eliminate the other 2, which I believe Tabitha was looking for. Spinosad, we're clarifying what needs to be measured for spinosad. Next. All right, so this is in the administrative policies. So we originally had a section in there about that the lab had to develop procedures for homogeneous portions that are going to be tested. What we didn't realize at the time is in the ag world that I come from, it's the biggest part once a sample gets to the lab is trying to assure that you have a homogeneous product before you start testing or pulling out portions. It was drilled into our heads. That isn't quite what we found here. So we're trying to clarify now with this portion of the rules that when we send out the information update in, I believe, October 2021, we were only addressing one aspect of all of this. And there's a little bit of confusion about that. What we were saying is you have to take out your micro sample before you take the rest of the sample and take it to the grinder that is not aseptic. That's what our information update says. It doesn't say anything about how the sample is prepped before that. So now we're clarifying to say all along, you should have been taking out a homogeneous portion for your micro sample, because that is what the science says. And then you have it aseptic, you're aseptically removing it. You also have to grind up or homogenize it in some way, that product. So the product that goes off for retesting, if it is not a homogeneous product, you're not sending holding back buds. What is being retested is the homogeneous sample portion that is being taken off for microbiology, because otherwise you're never going to have apples to apples when you do retesting, and you'll have a wide variation. So we're clarifying this. We welcome all discussion and comments. The second portion down here is we had a recommendation last meeting of the council of why can't we allow the sample retest to go to 2 laboratories at once. And that's what this is doing, is it doesn't have to go to one and then to another. So if a laboratory gets a positive and there's a request by the dispensary, they can send off 2 different samples. Next slide. All right, so
Steve.
Hey
What's
guys,
up?
so just some housekeeping things for everybody. So we have to make sure that the council members, you guys have access to that Q&A functionality. But because this is a public meeting, you have to make sure that you're not having discussions in that Q&A panel. Everything has to be done publicly. We have received some questions, so we will go ahead and read those in so that it's part of our public record right now. But we did just want to clarify that the main purpose of us showing all of these draft rule changes is just to make sure you're aware of them and how they related to the previous recommendations from the council. But because it's part of that rulemaking process, the best way for everyone, including our council members, to submit specific comments about this draft is to actually go to the rulemaking website, and I did send all of you that link, where there's the online survey, because that way you make sure that all of your comments are part of that rulemaking record that we go back and we actually respond to. So we are gonna go ahead right now and read all of the questions that we've received so far. But again, just keep in mind that the purpose of what Steve's presenting right now is really informational. This is the posted draft, so we aren't here to really get into the nitty-gritty of how things are worded and things like that. The appropriate place to make comments about that is really in the online survey that's open through either Wednesday or Thursday, I believe, this week. And Mariana, can you please read some of those comments that we've received?
Yes, so one of them is, why is an edible marijuana product and edible marijuana differentiated? So in other words, the phrase edible marijuana products and edible marijuana are two different things. They want to know.
Again, that's a comment to go ahead and put into the rules feedback, so that we can, sometimes we do make mistakes and things when we're drafting rules. I can't say that was one, but those are the kind of specific feedback that are more helpful for us to receive in the comments, so we can review them very carefully.
How commonly do mercury issues occur?
Steve, do you have any insights into mercury issues?
I know that I do not, because we don't see results. However, I do know that these four metals, arsenic, cadmium, lead, and mercury, were specifically chosen and are regulated by every state that I can see. I'm looking at a chart, because cannabis, the plant, is hemp is used on hazardous waste sites to pull up metals out of contaminated sites, because they know that these four are specific that cannabis will pull up. So if you have any of these four,
Steve, stop you for one more second again.
So council members, again, please do not add anything more into the comments or the chat or Q&A. All of those things, some of them are kind of off topic from our agenda and very specific items that would have to be added to another agenda. We have to make sure that we're complying with open meeting law. So we will read out all of the comments that are currently there. Please don't add anything additional at this time. We'll wait. We're gonna go through the comments that are there, let Steve finish presenting, and then if you have anything else you would like to discuss, just raise your hand and we'll do it verbally instead of adding to more conversations through the chat and Q&A.
so these four were chosen long time ago to be regulated. I'm not sure how prevalent the occurrence is in a regulated marijuana industry. That's probably all I have to say about that, because I don't know.
Steve, this is Max. I have a quick comment just to follow up. Right now there isn't a national survey on what contaminant there is in the cannabis that we have in the legal market, but this particular for toxic element contaminants of all the states that regulate elemental contaminant, all of them, there's no disagreement that this four should be included. So just to provide some backing on why this four.
Every state that I can see has these four. Some states have more, but all have arsenic, cadmium, lead, and mercury.
Okay, Daryl, can you go ahead and read the next question or comment that we have? We got to make sure we get all of these in the public record.
Okay, the next one is mercury within the state's level, and what product type versus another? What product type is experiencing more mercury problems than the other?
I don't think we have an answer to that, right, Steve?
No.
Okay. All right, Daryl, what is the next question?
The CAS numbers on the abacentin and Corinthians need to be updated too.
Okay, that sounds again like a comment that probably is not something that belongs on this agenda, so you might want to make that comment through our rulemaking feedback form or ask to have that added to a different agenda.
The next one is, how can you take an entire sample to 2 labs? Are the labs split off into homogeneous portions to submit the 2 labs? I thought it had to go in its entirety.
So again, those are some rule proposed draft language where if you go read the entire draft, Steve's just highlighting some sections here. If you read the entire draft language, I think it's a lot more clear about how that process will work.
The next one is a comment to that question. It says DBT sees mercury with edible chocolate at 0.2 off.
Okay. And the next comment, Daryl?
That's it.
That's all of them? Okay, guys. Again, please do not add anything else to the Q&A or comments. Raise your hand, council members, if you would like to. Oh, there's one more. Q&A.
Will we be adding a correction factor for absinthin and parentheses based on quantitating the entire product or basing the quantitation only on the listed components?
Okay, again, that sounds like a very specific item that would need to be specifically added to the agenda if we're going to discuss it. If there's a rule comment, go ahead and go to our rulemaking webpage. You can specifically make comments about rules you'd like to see changes to there.
Okay, we'll turn it back to you, Steve. Go ahead.
All right, so proficiency testing. So at some point, I believe it's still in the statute that ADHS is going to have to administer the proficiency testing program by January of 2023, I believe. But this is what we're proposing now, is that the proficiency testing samples need to be in a matrix similar to a matrix that's in medical marijuana or marijuana products. I think those are available now, unlike when we first started the program. And we are also taking out accuracy testing. In the beginning, we had to come up with a herbicide, which we no longer have to do. It took a while for proficiency testing companies to come up with products. I don't think that's a problem anymore. So accuracy testing is going away. Next. All right, so this will make a lot of the labs very happy. And this also addresses, I think, some of the issues that Tabitha was going to bring up. So in the beginning, we had very specific levels for the calibration standards. Now, all that's going to be required is that you have one standard at the limit of quantitation, and then 4 other standards for your 5 or 6, 7, whatever you need for your type of calibration model. So this is totally replacing for all the different testing, metals, residuals, potency. Potency really didn't have a maximum contamination level, but this is for everything. You now can build your curve at a level that includes the limit of quantitation. This is allowing a great deal more flexibility. I think the labs are highly capable of doing this. So that's what we're proposing here. Next slide. This is for the ICV. This is the ICV just needs to basically be a mid-level standard. There's no requirement for having it at a certain level. We also on here are allowing the ICV to have a slightly broader recovery range, 60 to 140 for propane and butanes. Next. The CCV, again, all it needs to be is a mid-level standard. Again, we are allowing broader range for the recoveries for propane and butanes. Next. All right, so there's a little bit more going on with LCS and LCS dupes. Again, you only need to do a mid-level standard. B is critical. The new B, they are spiked before extraction, no post-spiking. That is, we cited labs for that anyhow before because it's not scientifically valid process, but it's specifically being clarified in the rules. And then we have a number of different acceptance criterias that are basically the same. It opens up the criteria for propane and butanes to 60 to 144, or in each of the cases, you can do control charts. So the problem with control charts, if you're going to do it, is if you're a really good laboratory and you tend to always hit your limits between 70 to 130 or 60 to 140, what statistically happens is you will consistently tighten your control chart range to the point where you get so tight that you're going to have trouble. So we have a default limit that you can use. The problem that's out there is there are so many different kinds of products, and the current methods that are out there aren't necessarily a good method for testing what you all want to sell. So you may have a very poor recovery that's occurring with the product, but that's just how it is because that's the best science can do. What we're looking at with the lab control samples is that you have it under control, saying, here's the range of the recovery that we have statistically developed in the laboratory, so it's within control. Next. This is for matrix spikes, and so this will be a major change. We're proposing getting rid of matrix spikes for routine testing. While it may be the single most best QC sample for showing the accuracy of a specific dispensary sample, you don't have to currently do it on all samples anyhow. So you have to do it one out of 20 samples that are being run. If it fails, you are then supposed to qualify all 20 reports. And so the problem is the industry does not like to see qualifiers on their reports. So, and what does a failure on one product mean for all the other products that weren't tested for this? So what we're proposing is that anytime a laboratory is doing a new matrix, they run a matrix spike so they have an idea with that product type how well they can actually quantitate that sample. Next. All right, these are changes to the microbiology, not necessarily for what we're regulating on, but what we're putting into the rules. So the first one is for Escherichia coli or E. coli. And we're looking at having a limit of quantitation of at least 10 colony forming units per gram. So if you're currently using the Petri film method, and doing a 1 to 10 dilution, you do have a limit of quantitation of 10 colonies forming units per gram. Because one colony that shows up would equal 10. So we have some newer E. coli products that are coming out that have different ranges of accuracy at different levels. So even if you are doing a non-edible product, and again, if we do lower edibles down to 10, this is relevant, that you have a limited detection at 10. But for some of the newer products, we want them to dial in, similar to in the chemistry section, you have a limited detection that's lower than what you're actually quantitating at. Because if you're going to use 100 as your limit of quantitation with some of these new methods, the accuracy is very bad. So we want to have the dilution different so that it has an LOD or limit of detection at 10. I'm probably getting in a lot more than is necessary, but that's what we're proposing here. The next one is enrichment. It's currently something all of the labs are doing. And it's something that is a standard in the food safety industry. We at the state laboratory are involved with FDA food safety programs. They require enrichment. I have a slide at the end here that says we do not want to get too far away from what the FDA requires for their food programs, because if and when this cannabis is being legalized and probably run by the FDA, we don't want to be too far out of bounds from where the FDA is going to be. So every FDA program I have been a part of has enrichment. So we're proposing it. It's again something that is currently being required of all laboratories, so it's already being done. The next,
Steve, just a second. I just wanted to say, hope we seen your hand was raised. We're gonna wait till Steve finishes here, and then we can have you ask your question for the folks here. Okay.
okay.
Go ahead, Steve.
You said go ahead and complete the slide.
Yes, go ahead and complete
Okay.
them. I'll have questions after.
Okay, so number 4, what we're proposing is that there's a log that is maintained that shows samples that test positive or detected in the laboratory. Also, a sample is only retested. So what we're saying in B is if you run one of these and it shows up positive or detect, and all of the quality assurance has passed, and the instrument manufacturer, if you choose to go that way, says it's positive, then you have to report that. You can't just keep retesting it, trying to figure out whatever. So what B is saying is a sample is only retested when the quality control batch standards have failed or when recommended by the instrument manufacturer. And that's consistent with how we're regulating right now. All right, I'm done. So are there questions you said?
Okay, thanks, Steve. So we're gonna open it up for questions at this point. Hope, I know you had a question, so if you'd like to go first, please.
Thank you. I just wanted to clarify before asking the question. So if we have a recommendation, you want us to actually put this in the rules and not bring it up here? Am I understanding what you were explaining earlier, Megan?
Sorry, this room, we had trouble with the messages. This is actually a posted draft. A lot of the changes were based on recommendations from the advisory council last year. But at this point, because it's a posted draft on our website, the best way to make sure that your feedback is heard, and part of that rulemaking record really is to put distinct comments into that online survey. You can definitely, you can make comments and ask questions, but at this point, that's the best way to go about feedback.
Okay, then I'll just be brief and post it there. So I'm just concerned about 3. And so the reason why I am concerned about 3 is because I agree that enrichment are a standard, but I think it needs to be not a universal enrichment. And also, I think enrichment, when you say validated, and I would be happy to get into this more in a future meeting so we can discuss this in great detail, but we really do have to consider the enrichment bias that comes with many of these enrichment protocols. And maybe if it's just simply, if it isn't, why are you mandating enrichment if there are other methods? Also, I guess this is the second part of it, when there are other methods that detect without enrichment. And my biggest concern and why I bring this up is the competition. I mean, we're talking about days, 3 days for aspergillus, which is a common enrichment protocol for incubation. And it's often outcompeted if we're using a universal, for example. And so I just would really like to have some understanding and discussion about that, and whether that's here now or later, it's fine by me.
So to help, yes, go ahead and if any comments you're gonna make, do that on the draft, the online form. And then if there is something that you specifically want to talk about in the advisory council meeting, just submit it as an agenda item for next time. So this agenda, all we put was that we were gonna do the review, the overview by Steve of the changes to the rules that are in this draft. We didn't put anything on there that the council was going to be making any type of specific recommendations based on this draft. So if we're gonna be.
Thank you for clarifying that part, because I missed that fine print. Thank you.
So we would just need to know, and again, if everyone, takes the time to review the draft on your own, the whole draft, not just these pieces that are being highlighted. And then if there are things you want to discuss at the next meeting, we just need those specific items so that the members of the public have plenty of notice and opportunity to attend based on what's on the agenda.
Okay, any additional questions for Steve? Have you finished, Steve, or did you have any more slides here?
I have more slides.
Okay, you want to go ahead, please?
Yes. All right, so where do the qualifiers need to be on a report? So we have gotten in the past where we require that a certificate of analysis needs to be qualified. However, the qualifier can be all over the place and very small font hidden down at the bottom. And that's not the intent of what a qualifier is. So what we're putting into rules that the qualifiers need to be next to the data that it is affecting. That's what the first one is. All right, so we are now also dealing with changes that are happening based on changes that are coming in from the dispensary once a product has been submitted to the laboratory. So between the time that a product is submitted to a laboratory to the time it is being reported, if there's a change in any of the information in subsection B1A, then it needs to be documented on the certificate of analysis. And it may be a simple mistake or something, but it does need to be documented. Next. All right, so this is already should be what the laboratories are doing, because this essentially is the ISO standard 17025, which all the laboratories are required to be ISO accredited. So if a laboratory sends out a certificate of analysis and the dispensary or the customer requests a change, then this is the procedure. These are the procedures that are required in ISO for that changed, amended, or reissued report. I believe number 4 is from DHS though. Again, it's saying whatever is being changed must have something, the change is near where the change is being made, or it's flagged and footnoted so that it can be documented what is being changed. Next. Inventory system proposed changes. Okay, so the inventory in a laboratory is much different than a dispensary. In the dispensary, you have one unit. So you have a candy bar, and it's either in the dispensary or it got sold. And I don't know if they track it with the like stores where once it's sold, even the register can keep inventory through the system. At a laboratory, let's say that candy bar came in, it's going to be homogenized potentially. It's going to get apportioned out to many different parts of the laboratory and tested in different ways and possibly holding retest amounts. So it's not the same as a whole product. So we're putting into the rules the laboratories do keep control of all this, or need to keep control of all this, as with the current regs. They need to verify what is coming into the laboratory. It can't be the dispensary's inventory control. It's always, the intent has been the lab, once you take control of the sample, you have to start your inventory control. And so there's a verification. This is being done by most laboratories already. As the portions get taken out and to different parts of the laboratory, the LIMS system or control system in the laboratory, this is being documented. So when the product comes to the point where it's going to be disposed, the laboratory weighs up what is left, and the LIMS system or the computer system should be telling them at that point, here's all the portions that have gone off and been used for testing are gone. We should have 6 grams left, 7 grams left. At that point, that's when the dispensary will weigh the remaining product and be able to tell if there's possible diversion that has occurred or something's going on in the laboratory. So this is the main way a laboratory should be doing their inventory. There's also the process that you need to be able to pull up all of it at some point for the 30-day, or have it a LIMS system that can pull it up, because if you determine you have diversion going on in the laboratory, you're going to need to be able to pull up all of it onto a system to be able to tell what you've got at any point. So that's a change that's occurring here, hopefully to make it a little easier for the laboratory. Next slide. And so I'm basically done with some of the significant changes. I just wanted to bring up this article that just came out, because this is kind of what I've been saying. It's nice to see others that maybe have a similar view. What you will see in red is that once the FDA takes over this, she is saying, as I believe, that the testing are likely to align more with established testing industries for water quality or pharmaceuticals. And that these standardization of methods and associated method analyte matrix QC, a likely outcome of federal regulation, many pre-existing labs will need to strengthen their QC procedures. Now, I don't believe that's the case in Arizona. So what we have been trying to do in Arizona is to thread the needle. And based on our backgrounds, I've been associated, I have experience in a pharmaceutical lab. I have experience with FDA. I know how they regulate and how they want their lab programs. And so
This is Victor. We're running very short on time here, and I know we're not going to get to our items, so I'm just gonna have
Okay.
you there if you don't mind. Because we only have till 2:30 today. And I know we have a lot of items on our agenda to start with, probably too many. So what we can do and will do is add those items to our next meeting, but we do have still some time for Tabitha, maybe to Murray to maybe take one of their items of the three that they have left, and maybe we can have some discussion on one of those if you just want to follow into the next discussion on the recommendations of analytes to be removed and talk about the propane, etc. Or would you prefer to talk about one of the other two items that you have? I'll open it up to Tabitha and Murray.
This is Murray, and I'm going to take Megan's direction, and we're going to make a lot of comments online, including those things that we think are worth adding. And certainly Tabitha knows, or excuse me, Megan knows that I've got a lot of other hot buttons that I'd like to share with the team, whether it's through this venue or otherwise. So we're going to go ahead and yield to Tabitha and her points.
Okay, great. Thank you,
Thanks,
Murray.
Murray.
Okay, Tabitha, I'll turn it over to you.
Okay, hi, this is Tabitha. I don't think we have enough time to go over the batch definition slideshow that you guys have to flip through with me. So I think I'll save that for your next meeting, which do a follow up in a couple weeks, typically. Is that correct?
Yes.
Okay. I was just making sure it wasn't moved in the next 6 months or whatever. I was thinking it was to be a couple weeks, but anyway, so we'll go on to what some of the removal of the compounds that I wanted to suggest. Again, I brought this up at the last meeting, was propane. Propane's a residual solvent. We, in the history of the last 8 years I've been open, I've never got a detect for it. A lot of that is because it volatilizes at an extremely low boiling point is 42 degrees Celsius, negative 42 degrees Celsius. So pretty much you blow on it, it's out of your product. So I think it's a compound that we should remove. It would make it less expensive to operate for most labs that have a problem with propane, just because it volatilizes so fast, and just having that extra standard that we have to order as well. So I think it needs to be removed. Second one is acetylquinone, which is a pesticide. Another reason, my reasoning, and again, this is the one I brought up again last meeting, was it breaks down in water. So our samples are never going to have a detect for it because we water our plants that we grow. So this is a very complicated compound to run and to have actually pass most of the time because it breaks down so easily. And again, we're never going to see a detect for this compound. So these are the two that I'm suggesting. And then everything else was addressed in the rule changes as far as my other compounds that I had with the purethins and the curve. Everything else was addressed that I was going to go over, but those two are still remaining that I wanted to bring up once again. Hello?
Hello, yes, we can hear you, Tabitha.
Oh, okay, well, that was it.
Tabitha, do you have do you have that presentation regarding sampling and batch sizes?
I do. It's actually, we were gonna bring it up as a topic, and the actual slideshow they actually put on the website along with the agenda and all those other ones in the last slideshow. So if you click on that, you can see it. I mean, we can bring it up and browse over it, but I just don't think we have enough time for a full discussion to weigh it. Like, I think maybe we can review it and then bring it up again. Anyway, obviously batch is not defined in our rules right now as far as how much and what time frame. And so this creates a huge loophole in what needs to be tested, what size needs to be tested. And you can go anywhere from, you can call a whole room a batch versus one plant a batch. And so I think we're really jeopardizing our consumer safety by allowing not a more defined rule in our rules about what should constitute our batch size in Arizona. So with this slideshow in general, it's basically researching a few of the states that have had established limits for batch size, and most all states have something defined as far as what their batch constitutes. And I think we should take a look at this, review this, and adapt some kind of rule for Arizona that follows with the same trend as most states are doing. And it's for consumer safety. We're not right now with the loopholes. Not every strain has to be tested, and that's a problem. So anyway, I wanted to review this if we had more time to go over it and discuss some of these things. And the recommendations based on all the research that was put into this is basically comes down to, it's at the very end of this, but I put some recommendations that are pretty lenient and a good starting point as far as defining our batch size. And it's really just based on what all the other states are doing.
As a dispensary operator, I have intimate knowledge of cultivations that are bringing down 50 to 400 pounds of one strain, and they're doing one test. One test, and that clears all 400 pounds from mold, pesticides, potency, everything. This is not what this panel and this testing law was intended to overlook. I think we need to have a good look at this.
So just to make sure we're compliant with open meeting, there was another comment added that said, for form on reducing the amount of time for labs to be able to present. Labs have a large number of issues that need to be addressed. Again, we are not trying to reduce the amount of time that the labs are going to have to discuss these issues. We just ran out of time on our agenda today. We had too many requests to add items to the agenda for the time that we had given. But we intend to meet monthly, probably for the rest of this year. So the things that we did not have a chance to fully discuss today will be up first on the agenda next month. And all of these presentations, including this one that Tabitha was sharing with us, is available on our website now. So everyone will actually have plenty of time to review it before the next meeting. So hopefully that will lead to even better discussions during the next meeting.
Yeah, I think the general concern is just that, and I get it, you guys have a lot going on. This is Ryan Tracy with C4, by the way. Is that we just don't want to see it kicked. We thought we were going to be meeting in the beginning of the year. I get it, stuff gets in the way, and then here we are, July. And I think to Steve and Tabitha's point, this is a glaring hole and something that has to be addressed. I mean, with the intent of the program is being circumvented and subverted by a lot of different folks out there, and it really is hopefully won't catch up with us and get somebody sick or have an issue with consumers. But to Steve and Tabitha's points, it is absolutely rampant.
Absolutely, we will discuss this again. We will have this on the agenda first, one of the first items on the agenda next month. I do want to point out we did have, was a recommendation of the advisory council last January. The council actually recommended many things that were outside of the purview of the council in the January report, including having different types of technical assistance sessions outside of testing, which the department has done all throughout the beginning of this year. We pretty much had monthly technical assistance and work group sessions, and there were several related to batch definition and certificates of analysis. But we will also continue to have those conversations here with the advisory council.
Is there a way that the members of the advisory council can be a part of those technical meetings, or is that just something that's done internally with you guys?
Those invites went out to all licensees of both dispensaries, establishments, and the labs.
Okay. I'll have to look closer.
Okay, thanks for your comments, Ryan. Murray, you had your hand raised?
Thank you. I appreciate you recognizing me. I think you that Megan answered the question, which is when will we be meeting again? As a consequence of this meeting, will we be able to set a date, or is that something that'll just basically be on the state's calendars and you'll inform us?
We will have to coordinate here to make sure that we have the room and the people available to help coordinate the meeting, but we will get that out to you as soon as possible for September.
That's great. Thank you.
Absolutely.
Tabitha, did you have anything additional to add?
No, we don't, like I said, we don't have time to discuss it. I do want to go back to the points, and I don't know, since I ran through them about the compounds to be removed, really retouch on that and get everybody else's opinion, especially from the other labs that are on the phone right now, since we just kind of ran into this really fast, because I know a couple people wanted to discuss this part of it, but I'd like to keep that on the agenda. So, I mean, it's already on the radar. I think a brief discussion about those two compounds in particular would be ideal in the next meeting as well. If we could revisit those, as well as start the discussion next time about the batch definition.
We will absolutely make sure those are on the agenda. And if anyone has any data or any supporting documentation for those discussion points, please feel free to send those our way as well. That would be helpful.
And Tabitha, you'd also want the discussion on your recommendations of requirements for maximum allowable concentration on the next agenda also?
Oh, no. That was pretty much addressed already with the revision to the rules, or the draft, I guess, with the curves, and it has to do with metals and stuff. It's fine. I'm happy with the rule changes that you guys took care of already.
Okay, got it. Thank you.
And Murray, I think you and I can probably reach out to make sure I know exactly what you want to have on the agenda next time as well.
That'd be great. Thank you.
Okay, thank you, everyone. I appreciate you being accommodating to our situation today. And we will, as Megan says, get those on the agenda next time. I'd just like to ask if there are any discussion again of any additional items that need to be discussed today, in addition to what we've already talked about, as in additional items that you want to be on the next agenda.
Should we just email that to you? I do, and I just assumed I'd be emailing it to you.
That would be great. We always do send out, a couple weeks before the meeting, a call out for any additional items you'd like to have on the agenda. Just keep in mind, if we have a ton of items, some may get pushed to the following meeting. It's just a time constraint, but the more specific you can be with what you would like on the agenda, that's extremely helpful. And if you are going to be leading a topic, any type of documents that you would like the committee to review, if you can provide those ahead of time, we can make sure they're posted and sent out to the council members as well.
Thanks.
Okay.
And Megan, historically, when we do these meetings, it's always been the department presenting for the first 2 hours and 20 minutes, and then, we get to discuss the issues we're having. Can we get some type of confirmation that, the things that were missed today, that yes, those take front of the page, those take front of the line, we will be discussing this. We will not be, looking at other things first.
So typically, yes, we already went through Steve's.
I know, I'm just verifying it.
So probably the only other thing that would be before these items would just be, you guys had also requested that we give you the top deficiencies for dispensaries, establishments, and labs. So we would probably go through that very quickly at the beginning.
Yep.
And then add the items as we have said we would do.
Thank you, Megan.
Yes.
Okay, well, I think we sort of wrapped up in terms of what our next items are and what will be coming up on our next agenda. And again, if you've got items, please feel free to email them to us. We appreciate all of your input today and for attending the advisory council. And again, feel free to reach out to us if you've got any questions for the next meeting, and we will work on getting that set up for September and notify all of you of that. Again, appreciate your time for being part of the council, all of your input, and I'd like to finally call for a motion to adjourn.
I second.
All those in favor, say aye.
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 Aye. Aye.
Okay, thank you, everyone. Meeting adjourned. Hope you have a good rest of your day. Thank you. Bye-bye.